everyone, I'm Brendan Farrell, a postdoc here in the Higgins Group at the Biochemistry Department at Kavli Institute here in Oxford. Today I'm here with my co-authors Melissa and Norsad, and we're here to talk about our recent paper published in Nature, which describes the structure and function of an essential protein complex which is involved in the invasion of malaria parasites into red blood cells.
So malaria is a really deadly disease that impacts almost half the world, and it's estimated that there are over 200 million cases per year, and this results in almost 600,000 deaths, particularly in sub-Saharan Africa, where children under 5 are typically the most affected. And so it's really essential that we are able to develop a vaccine. There are some in the pipe works, but we really need a very effective vaccine that will stop parasites from spreading from person to person. The type of vaccine that we're looking at is geared towards the part of the parasite's life cycle where they infect human red blood cells. And so malaria parasites, when they infect people, they are injected into the human host by the mosquito. And then these parasites will travel to the liver, where they'll hunker down there for a little while and multiply. And then finally they're released into the bloodstream. And then they begin cycles of actively invading red blood cells, growing within them, multiplying, and then bursting out and beginning this process all over again. And so if we can stop red blood cell invasion from happening, then we can stop parasites from multiplying and we can stop disease symptoms. And so vaccines aimed towards the blood stages will help us to achieve this. Erythrocyte invasion is a really complicated process, and it's quite a cool thing to look under the microscope. And when you watch parasites live invading red blood cells, we can see that it's a multi-step process that begins when individual parasites bump into the red blood cell and then they move across its surface. And as they're doing this, they're engaging with receptors on the host cell surface. And Rh5 in particular will bind to bathogen on the red cell surface, and this is a really essential step of invasion that occurs just before the parasite starts to burrow itself into the host cell. And without Rh5, so if you conditionally delete it from parasites, parasites aren't able to invade. This video here is showing the invasion process. And what's happening is you're seeing parasites bounce along the red blood cell and deform it, wrapping it around them like a blanket. And then the green signal that you're seeing is coming from a calcium-sensitive dye. And without Rh5, that fusion process can't happen. So it's a really essential protein complex that we're working with. And if you can target this process and stop it from happening, then you can stop invasion from happening.
So this essential protein complex in Plasmodium falciparum is composed of 5 proteins, and all 5 of these proteins are essential to invasion. In fact, you can generate neutralizing antibodies or nanobodies to all of these. And while our group and others have structures to some of these components, we don't have an idea of what the entire thing looks like at high resolution. And so what we wanted to do was solve a structure using cryo-electron microscopy of part of this complex, the RCR complex, which has 3 proteins, RH5, CYRPA and RIPA, so that we could see how these proteins interact and what they look like in higher detail. So in this technique, we take our sample which we've purified, and then we suspend it in a thin layer of buffer which we can flash freeze, which we can then image in the electron microscope. And this gives us projection images of our sample in 2D. We can then take these and then reconstruct them back into 3D such that we can see what this protein complex looks like. And what we can see now is that how these 3 proteins, RH5, CYRPA and RIPA, interact with one another. And for the first time we can see RIPA. And what we can see is actually it's quite complex.
The structure which Brendan has solved provides a really clear snapshot of this really large complex. Proteins are quite flexible molecules, so they can often cause significant structural changes during the biological processes. There are multiple key players in this complex, and we wanted to do a bit more deep study to understand the functional aspects of different components of this complex. Rh5 is the prime candidate for vaccine development, and initially we focused on Rh5 to do those functional studies. There are a few proposed models in the literature about how this complex might be working, and there are models which talk about significant structural changes in Rh5, and we haven't seen such structural changes in our studies. So to test those hypotheses, I have used protein engineering approaches to model more stabilized form of Rh5, which will inhibit such structural changes, and to see if the parasite with this engineered version of Rh5 is capable of invasion or not.
So what I was able to do is I was able to take the information that Norsad had provided and replicate his findings in the parasite. And so using CRISPR-Cas9, I was able to generate a parasite line that had this locked version of RH5 that wasn't able to undergo the gross structural changes that it's been predicted to do. And then what I did is I looked firstly to see could the parasite survive, because if this locking mechanism was going to disrupt invasion, then obviously the parasites couldn't grow. And then the second thing I was able to do was look using live cell imaging to visualize the individual steps of invasion and to determine that everything was normal.
So Norsad and Melissa's work nicely demonstrated that RH5 probably is a rigid molecule during invasion, and what this really highlights is just how little we understand actually about the invasion process. But RH5 doesn't act alone, and if we zoom back a little bit, the RCR has 3 proteins, and in fact recently we found out there's 2 more proteins that are in this complex, CSS and pTRAMP. And these act as an anchor for the RCR complex to the parasite. And so with our new structure we then wanted to address, well, how does this all actually fit together? And while RCR is anchored to the parasite via these 2 more proteins, we didn't know how this happened. And our structure enabled us to discover that it's actually through this tail of Ripper, which is this flexible, elongated arm which comes out from the core RCR complex. And it's with this tail that the complex is anchored towards the parasite. And the fact that this part of the RCR complex is not visible in our EM structure suggests it's attached flexibly to the rest of the complex. And what this means is that this could have functional consequences, but really it means there's many more questions that we need to really address. Now that we have this, say, broader picture of what this large complex may look like, we're in the position now to look at how antibodies which are generated in vaccination actually attack this complex and block the binding to bastion from happening.
So in this study, we have learned quite a bit about how this really large complex works, structurally as well as functionally. We have a long way to go to fully understand the implications of different interactions in this complex. We can definitely look more into how the antibodies against Ripper works, how the CSS pTRAMP is interacting with the rest of the complex, and what are the implications of such interactions during the invasion process. The structure-guided approach which you are utilizing will have the potential to design
next generation vaccines.